Osmosis can be defined as movement of water through a semipermeable membrane. Semipermeable membrane are barriers to water movement because they allow solvents, but not all solvents, to pass through. Although water moves back and forth through most semipermeable membranes, it flows more forcefully from the side of the membrane with lesser solute concentration to the side with greater solute concentration.
Cell membranes are semipermeable and are therefore affected by osmosis. In an isotonic solution, the concentration is the same on each side the cell membrane, and the cells retain their normal size and shape. In a hypertonic solution, the concentration is higher outside the cell membrane, and the cell shrinks because the osmosis pulls out water. In a hypotonic solution, the concentration is higher inside the cell membrane, and the cell swells because osmosis brings water in.
Osmosis is important to consider in the medical field when storing red blood cells. A hypotonic solution has less sodium than that of the patient's currently circulating plasma. It would be used to push fluid from the vascular spaces into the cells. These are normally kept in a plasma solution, which is isotonic to the cells when it contains specific proportions of salts and proteins. However, if red blood cells are placed in a hypotonic solution, or one with a lower solute concentration than in the cells themselves, this can be highly detrimental. In fact, the plasma solution used by most hospitals for storing red blood cells is slightly hypertonic relative to the cells to prevent them from drawing in water and bursting. Physicians use a similar solution when injecting a drug intravenously into a patient. The active ingredient of the drug has to be suspended in some kind of medium, but water would be detrimental for the reasons discussed above, so instead the doctor uses a saline solution that is slightly hypertonic to the patient's red blood cells.
Today we're going to do an experiment to show how cells react in all three different types of solution. In this experiment, you'll need 3 hard-boiled eggs, 3 different types of solution, salt water, which represents our isotonic solution, right here, corn syrup, which represents our hypertonic solution, and water, which represents our hypotonic solution. And we also color dyed it so you can tell the difference. Here we have our cups filled, and before you drop the eggs into each cup, you would want to remove the eggshells. So I drop the eggs into each cup. And with the corn syrup, it actually doesn't sink, so you have to use a spoon to push it down. We will let this sit for 48 hours and see the result of the eggs. It's been 48 hours and we took out each egg from their solution. The first egg, which was in the isotonic solution, as you can see, the egg is actually the same size because the egg and the water have equal solute concentration. The second egg was in the hypertonic solution, which had more solute in it than the egg. Therefore, the egg shrunk. This is because the water moved from the egg into the solution. The concentration is higher outside the egg, and the egg shrinks because osmosis pulls water out. And here we have our third egg, which was in the hypotonic solution. The egg actually increased in size because the solute concentration was higher inside the egg, so the egg swelled and osmosis brings water in.